Hey guys, welcome back to Science with Shane. So today we're going to be talking about tyrosine kinase receptors. So tyrosine is an amino acid, one of 22 types, necessary for cellular protein synthesis. A kinase is an enzyme that phosphorylates its substrate. It catalyzes a reaction between its substrate and an energy carrier molecule like ATP, resulting in the ATP losing a phosphate group to react with the substrate to form a product that is highly energized. Tyrosine kinase receptors are transmembrane proteins, right here, that are integrated into the plasma cell membrane, reacting to ligands on the extracellular side and substrates on the intracellular side. The base structure is a protein that hosts tyrosines on the intracellular side and a receptor on the extracellular side. Once fully activated, RTKs trigger many important cellular functions such as growth, cell differentiation, mitotic cycles like reproduction, endocytosis, and even negative effects like the creation of cancer cells. So prior to binding with a ligand on the extracellular side, an RTK exists as a monomer of tyrosines with a receptor. The first step is the ligand, the signal protein, this blue guy right here, binds the receptor. This results in dimerization, the binding of the ligand that brings two receptors together. So they're together now. The receptors, which are tyrosine kinases, can now phosphorylate each other. The dimer then takes on the intracellular side, which means the ATP over here donates a phosphate group, resulting in ADP because it lost a phosphate. And this process happens 6 times, allowing 6 ATPs to be attached to the intracellular side of the receptor. So once this occurs, you can see that the relay proteins get activated and phosphorylized through the tyrosine kinase. So these are the relay proteins right over here. We're going to take these, and we're just going to shove them in.
Please, it is a Ratner, Alaire Rowe, Austin Rosario.
My relay protein.
Jose Palacio.
Once these relay proteins are activated, they are able to carry out a cellular response by leaving. Right here. Okay. Turning on the genes involved in growth and cellular differentiation. All these great cellular responses that can occur now that these relay proteins are activated. Now, our specific example is endocytosis. Now, in endocytosis in a eukaryotic cell, tyrosine kinase receptors catalyze a reaction between the substrate dynamine and the phosphate groups taken from ATP molecules. Dynamine is a GTPase, which is a special type of enzyme that catalyzes hydrolysis, the breaking up of chemical bonds by the addition of water. Dynamine's reaction with the energized phosphate groups results in a product that induces scission in the cell membrane, the budding of a vesicle that transports the substances entering the cell. This product also works with a special protein called clathrin and adapter proteins to create a clathrin-coated vesicle. Where on the plasma membrane clathrin is not present, RTKs often trigger endocytosis as part of a flexible combination of lipids in the cell membrane dubbed a lipid raft. Once the endocytosis are transported into the cell to where they are needed, since RTKs were part of the transport vesicle, they can now themselves be degraded within the cell or recycled back into the cell membrane. Thanks for watching another episode of Science with Shane.